Good morning. My name is Sarah Jana, CEO and co-founder of My Intelligent Machines, and that's a pleasure for me today to present you the results of the study we made in collaboration with the Department of Computer Science of the University of Quebec in Montreal, and that aims at identifying new drug targets controlling metabolic reprogramming in triple-negative breast cancer. The rationale of this study was really that there is different studies showing that metabolic reprogramming in triple-negative breast cancer and basal-like breast cancer and also cancer stem cells is associated and contributes to chemoresistance, metastasis, and disease relapse, and results in poor outcome for patients. So the aim of the study was really to identify new genes controlling this metabolic reprogramming of TNBC. And for that, we developed a new methodology based on MIMs, infer genetic interaction graph. So briefly what we've done is that we retrieved transcriptomic and clinical data from breast cancer patients, 327 of luminal A and 204 of basal-like. And using this transcriptomic data and also our MIM site ontology, which is composed of knowledge coming from the literature that we extract using natural language processing, as well as knowledge coming from 120 public databases, we are using our patient GI2 algorithm, which is inferring for each patient between 1 million and 2 million genetic interaction, and results in the generation of genetic interaction graph that are shown here. Then with this graph, we have different steps. So the first step was to identify consensus graph for basal-like patients and a consensus graph for luminal A, and to identify a subspace within this graph that are genes that are interacting at least with one gene associated to OXPHOS and one gene associated to glycolysis. And we already shown that the subspace of the graph are enriched, significantly enriched in genes that we identify as Warburg effect genes. So these Warburg effect genes are extracted using our NLP capabilities from the literature, and there are genes that have been associated to Warburg, and that were divided in training set and testing set. So based on the graph, this subgraph, we're extracting two kind of features. We're extracting expression-based features associated to differential expression, correlation of expression, etc. But we're also extracting GI graph-based classifiers, attributes that are based on topology and also values of different values of the graph, including also the expression attributes. And with all these attributes, we are training two different kind of a classifier based on expression attribute only, and classifier based on GI graph topological and value, including the expression attributes. And then after we're training this classifier and testing them on a testing set that is composed of genes that I described earlier. So in this slide, you see the results and the performance of the different classifiers. So you have a precision recall curve and also ROC curves that are indicated. And on the left panel, you have the different AUC of classifier that are trained on luminal A subspace, Warburg subspace, and basal-like. And we trained 5 different classifiers, are shown here only the results of the one that worked the best for both expression and GI graph. And what we've seen, so you have AUC for GI graph-based classifier, expression-based classifier as a control random classifier. And what we can see on this slide is that the GI graph-based classifier outperform the expression-based classifier that shows the value of using graph-based methodology to train this classifier. So from this classifier, we identify the genes with the top 5 highest score in Basal-like classifier, and we ended up with a list of 85 genes. And from these 85 genes, then we identify genes that have the highest probability of being good target. And for that, we first of all select the genes that have low pleiotropy, so meaning that genes using the UK collection are associated with functional alteration associated with no disease, no trait, associated with that are considered as trait specific or domain specific, but not excluding multi-domain genes that are high pleiotropy genes. And this actually aims at trying to reduce side effects of drugs targeting this target. The second selection we did is by selecting genes with high degree within our GI graph, which means that there are genes that are connected, genetically interacting with a lot of genes. As shown is in the graph on the left, this is a cumulative distribution of degree for drug targets in green, biomarkers in orange, and overall graph in white. We showed that drug target in breast cancer tends actually to have high degree. So we select these genes, and then we ended up with a list of 17 targets using this approach. And what we've observed is that 3 of them are already at clinical stage development for breast cancer and TNBC particularly. And there is here, which correspond to high enrichment with the odds ratio reaching 124 with a p-value which is quite low here. A second selection that we do to identify therapeutic targets is predicting whether these therapeutic targets will, when they are targeted with a drug, will have like a high response rate in basal-like tumor with respect to luminal A. And for that, we're using a methodology that we developed for indication prioritization that consists in using each graph of each patient to predict whether this patient will be a good responder to a drug, knowing only the name of the drug targets. So this methodology actually use the mean z-scored expression of the genetic interactors of the target. And we've shown that this mean z-scored expression has a higher potential to predict good responders to a drug than the z-scored expression of the target itself. And this is shown in this graph for sorafenib on a cohort of hepatocellular carcinoma patients, and we show in this rock curve here high prediction with when using mean z-scored expression of genetic interaction that we call in this case focus genes. So using this methodology, we assess for every of the targets that we identified whether they have a highest expected response rate in basal-like versus luminal A. And what we've shown here is that we identified 8 targets that has a very high response rate above 70% in basal-like and much lower expected response rate in luminal A. And from these 2 of them are targets that are in clinical development for breast cancer and TNBC, which is PARP1 and AKT1, which actually is highly enriched. And you have here the odds ratio reaching 186 and also the p-value. What we observed from this list of genes is that they are highly enriched in signaling proteins, and they are also connected to glycolysis, as it doesn't really show in this graph, but there is actually on metabolism only glycolysis that is enriched here. So in conclusion, we develop a new methodology for target discovery based on patient GI graph. built using MIMs patient GI2 algorithm from tumor transcriptomic and transcriptomic data and MIMS site knowledge base. This methodology identified 8 potential drug targets, which are signaling protein that we predict to control metabolic remodeling in basal-like breast cancer, 2 of which, PARP1 and AKT1, being already targeted by drugs in clinical trial for TNBC. So the future of this project right now is like validating this prediction, and the drug targeting this protein, the protein and identified genes will be tested for their antitumoral activity in 3D organoids from resected TNBC, a tumor, and PDX mouse. And please do not hesitate to contact us for any questions you might have, or if you want to participate to this project. Have a nice day.